So these are the lessons learned. I think chimerization is highly attenuating and maybe over-attenuating for some viruses. Again, for live vaccines to induce immunity, we have to infect and replicate, and we need balanced homotypic immunity. And that's been very difficult to measure because we have cross-reactive antibodies. And Gabriela hosted a meeting at CDC Puerto Rico just a few weeks ago, trying to say, can we come up with an assay that can distinguish between protection and risk with these dengue vaccines? And that's something I think that we really have to work on. And there were a lot of very smart people in that room who are working on that now. I do think that T-cell immunity is important for mitigating disease, but of course it's very difficult to have a standardized assay to measure that for support of immunity. So other considerations, and I think we're going to talk about some of this in our panel discussion, is how are we going to ever have enough vaccine to meet the global demand? The global demand is only going to grow, and I think we only can supply a minute fraction of that right now. There are additional manufacturers that are going to come online in the next 3 to 5 years, but that's still 3 to 5 years away. That's not tomorrow. And then how do we integrate these vaccines into a vaccination schedule that can facilitate uptake? For some of these vaccines that require 3 doses or 2 doses and ages in adolescence or higher, that may be difficult. And then really importantly, how do we monitor? How do we follow for long-term safety signals? I'll end it.
So I think we have time for 1 or 2 questions before our panel discussion, if there are any. Yes. Gabriela, please.
Why do you use a chimeric virus for the dengue 2 conformation? That's one question. The second quick question is 50% efficacy for dengue 2 among seronegatives enough? And what about that 50% that doesn't have protection?
So,
Would you have enhancement?
So,
Can you repeat the question?
yes, so the first question is why do we use a chimeric? And that's a great question. Early days, we tried many, many different components. And we, as I said, we looked at them individually. And the NIH's original strategy was to have a chimeric dengue 1, 2, 3 on dengue 4. But the chimeric dengue 1 didn't work. The chimeric dengue 2 in non-human primates looked attenuated but still induced an immune response. So it went forward into human studies. And then in human studies as a monovalent, it's still, although it infected, we had about 80% seroconversion with that. So we moved forward with that. We had a 3-4 chimeric, which was over-attenuated and was tossed. And that's what prompted different dengue 3 candidates, including the dengue 3 delta 30, which was under-attenuated, and the dengue 3 delta 30, 31. And I said to Steve, I think, if we had that 10 years ago, we may do a dengue 2 delta 30, 31, but it was too far advanced. And we do think the second question was, is 50% efficacy in seronaives at 3.7 years, is that enough? And I think against virologically confirmed dengue, it is if you have, again, homotypic immunity. So I think what was more compelling or encouraging was at 3.7 years seeing almost 90% efficacy against dengue with warning signs and severe dengue, meaning that even if you're not preventing infection, you're preventing the more severe complications. But that's something that's going to have to be followed. And it'll be interesting to see if there are second-generation vaccines that come down the pike and they replace that. There's other concerns about even different strains and strain variation and how will that affect efficacy and things like that. And I think we're going to continue to learn from dengue for many, many, many years after these vaccines get deployed. Maria.
Hi, so interesting, totally not my field. But in my field, we have indirect effects. And so I'm really curious, if you reduce infection, can you reduce transmission within the household or otherwise?
That is a great question people are looking at. And one of the questions is, do we want to completely reduce transmission? Because we may get natural boosting, which could provide additional protection for many, many years. I think where there is greater indirect protection, and it's something that I alluded to earlier, is if you can just prevent hospitalization and severe disease, I think you're making a great contribution. And there was a lot of debate about both Dengue and still debate about TAC 003 with that. I mean, Dengue could have had, I think, a huge impact in terms of hospitalization and severe disease in the most affected countries. But you have that ethical dilemma of when you've identified an individual risk, are you then compelled to avoid that risk? And the answer was yes. So now we have to test everybody before they get vaccinated. And Gabriella will tell you that Dengue uptake in Puerto Rico has been terrible for many different reasons, right? It's just been very, very low. And there's some, and you can confirm this, that Sanofi may stop making if they haven't stopped producing Dengue because uptake is so low.
Yes, I have a question for you. So thinking about going back to thinking about the fact that dengue 2 is chimeric in this vaccine, you spoke a lot about how you think that the T-cell response is important for immunity, and that the T-cell response is predominantly to non-structural proteins of dengue. So do you foresee there being any issues down the pike with homotypic immunity dengue serotype 2 because it's on a dengue 4 background?
That's a great question. I have so many slides I can start. But, so we did a study where we looked at the CD8 T-cell responses to individual monovalent components and the CD8 T-cell response when you give it as a tetravalent. What's really interesting is when you give a monovalent, most of the CD8 T-cell response is directed against serotype-specific CD8 epitopes, so those individual. When you give the tetravalent, that response moves to conserved epitopes across each of the 4 serotypes. So the tetravalent, about 80% of the CD8 epitopes that were recognized by after tetravalent vaccination was conserved across all 4 serotypes. So I don't think it'll be a problem because we have the 3 other serotypes. We did other experiments, but I'll tell you later.
Thanks, Sam.
So that's great, and I think what we're going to do is move to our panel discussion. So I'm going to ask Anna and Gabriella to